The title of my talk today is Metal, Mobile or Stationary to Enable Catalysis. And I would like to discuss this with an example of aldolasers that we've recently been busy with. And aldolasers are grouped by the donor. That is the molecule they activate to act as a nucleophile to attack the acceptor or the electrophile. So this is the electrophile. We've got 4 possible nucleophiles, and one I'd like to concentrate on is pyruvate today. And this is something to do with a synthetic target that we had a few years ago and still have. So we wanted to make this type of compound. And we argued that you could make this bond here with a pyruvate-dependent aldolase if that pyruvate-dependent aldolase would accept an hydroxypyruvate, so a hydroxy keto acid. And fortunate for us, another group solved that problem for us because they did a major screening and they found lots of these enzymes. So a French group some 7 years ago published lots of these enzymes, amongst them the one from Single-Monas Wittichi and from Bocalderia phytoformans. And that are the 2 enzymes I'd like to focus on today. Now, they accept hydroxy keto acids, which is why we called them then hydroxy keto acid aldolases, HKA. And the French scientists said they need divalent metal. And with this hydroxy group, when you perform the reaction, you set up 2 stereocenters. So you overall have 4 diastereoisomers. And what they could show is that the stereocenter you set up out of this carbon always points forward. And the SW, this stereocenter points backwards. And for the BP one, it points forward. But the stereo control for C4 is not perfect. That's a different story. I'm not going to talk about that today. Okay, so we then produced these enzymes. We wanted to characterize them, and we had a good look at the literature, and a number of pyruvate-dependent aldolases had been characterized. And for the class 2 ones that require a Lewis acidic metal, a mechanism had been proposed. So you've got the magnesium sitting here, and we've got the pyruvate coordinated to it with 2 ligands. So that's basically what you see here. So you've got the metal and the pyruvate with the acid group and the keto group associated to it. Then the base here deprotonates the water coordinated. You create your enolate, and this arginine, that is this arginine, puts the aldehyde in place, and then you can get a nucleophilic attack of the donor on the acceptor, and then you get the new carbon-carbon bond. And as these enzymes are also in nature degrading enzymes, the mechanism is here drawn for the degradation reaction. Okay, so this is the case for the pyruvate-dependent enzymes, and we had a hydroxypyruvate, so we were wondering, is that all the same, and why does this enzyme, or why do these enzymes also accept hydroxypyruvate? So, which metal do they use? How does the hydroxypyruvate coordinate? Is that the same way? Is the arginine still important, and what's the base? So let's find out. The first and easiest thing was to look at the metal. We produced the APO enzyme. We added different metals, and you can see that manganese or magnesium for both enzymes are the favorite. And where's it? Copper didn't do the trick, right? No. So I have to disappoint you, but it's also not a redox reaction, so that's okay. Huh?
It's not work all the time.
No. So anyhow, good. Then we wanted to know about the coordination. Whoops. And I don't know why these got smaller, but that was not intentional. So we looked at EPR spectra, and you can see the SWHKA, and you've got the characteristic spectrum of a manganese 2 plus, and a little bit of a bump here. If you add hydroxypyruvate, that bump gets larger. If you add pyruvate, you get a completely different EPR spectrum, so obviously a higher order spectrum. And if you have pyruvate, whether it's in the presence of phosphate or not, there's no difference. And I'll get back to that phosphate a bit later. For BPHKA, so this is the just manganese 2 plus. This is the enzyme with the manganese 2 plus. If you add pyruvate, we get a little bit of a broad spectrum. And if you subtract this one from this one, you get the same spectrum as you can see here. And as this is not quantitative, we can't say whether this is 10% and this is 90%, or this is 90% and this 10%. Good, and again, that are the spectra here. Phosphate present or not doesn't make a difference. Good. So we also did X-ray crystallography, and there you can beautifully see how this hydroxypyruvate coordinates. It's exactly the same way as the pyruvate does. So you've got the metal and the acid group and the other keto group here coordinating, and two amino acids, two acidic amino acids coordinating the metal, and then we've got two water molecules coordinated. What we found out was the key to the ability to accept hydroxypyruvate was that we have a phenylalanine here, and the enzymes that don't accept it, this is typically a very apolar amino acid. And the same was true for the BPHKA, and here again you can see the metal, the hydroxypyruvate coordinated, and the two acid residues coordinating the metal. And if you do an overlay of the two structures, then you can see that they're virtually identical. So we now know how the thing coordinates, so that's nice. What about that arginine sitting here? If you mutate it away, you lose all activity. And we also then did X-ray structure analysis where we soaked the sample with D-gluceraldehyde in addition to the hydroxypyruvate. And you can see it located here, coordinated to the arginine, setting it up in the right way for the reaction. And now, what I said earlier, the C3 of hydroxypyruvate gives the stereocenter C3 of the final product, and this here gives the stereocenter for the C4 of the final product. So here we've got an enolate, and the enolate is always formed like this. The hydroxy group never points this direction and attacks with this side. So this is why you've got absolute control over the stereocenter. And the attack can take place here, but this can coordinate in two different ways, and depending on how it coordinates, you get different stereochemistries. So for a chemist, you get coordination, you deprotonate, you only get one enolate, which attacks with this carbon always on one side, and it can attack the aldehyde here either like this or like that, and depending on how it attacks the aldehyde, you get different stereochemistries here, while this one is always perfectly preserved. Okay, so glutaraldehyde docks. If we mutate away our histidine, we again lose activity, so that again clearly is the base doing the job. We then looked, of course, at different buffers and which buffer is the right buffer to use. And when we did this, we had a bit of a surprise when we used the SWHKA, because here we had a very high activity in the presence of phosphate. And with all the other buffers, the activity was not so high. For BPHKA, there was not such a difference whether you use phosphate or the other buffers. Actually doesn't really matter. And there's a small difference, but it's not really significant. And of course, we wanted to understand this. But first of all, we wanted to be 100% sure that it wasn't a pH effect. So we set the pH with bis-tris buffer and added a little bit of KPI to it. And then we got exactly the same data for activation. If you add too much KPI, you actually precipitate your metal. And this is something I can remind all of you very much in general. If you've got a metal-containing enzyme, check whether your buffer precipitates your metal and don't add too much buffer in that case. Because I think that some people do make this mistake, and we only noticed that we could have made the mistake ourselves due to this experiment. Okay, good. So we then wanted to check what is actually really accelerated so much and how does this really work. And if you look at the overall mechanism, then we first have to deprotonate. So we have this step that we can accelerate, and we can accelerate the second step, which is the carbon-carbon bond formation. And you typically analyze it via a carbon bond degradation. So when we did this, we looked at the deprotonation in a simple NMR experiment. You take your hydroxypyruvate or pyruvate or ketobutyric acid, you put that in D2O, you add the enzyme, and you follow the HD exchange. And then we saw a 120-fold increase in rate if phosphate was present. If it was absent, a lot slower. And this was actually also described in the literature for a few of these pyruvate-dependent enzymes. If we look at the second step in the reaction, so K2 or K-2, we analyze this with this synthetic reaction where you cleave off CO2 and then you get pyruvate, and the pyruvate you can reduce, and then you can follow the NADH NAD in the UV, and via that you can directly analyze. And we saw a 10-fold increase. So we wanted to know what the phosphate does and where it goes, and we try to get the phosphate into our crystals. And I think we tried for 2 years to get phosphate in there. Then we gave up, we submitted the paper, and then one of the referees said, please dock it. So then we decided, okay, we make a model with the docked phosphate. So this is the docked phosphate. You can see the phosphate is in a position close to the pyruvate to aid the deprotonation. And you can see here this pink side chain of the SW enzyme. If you overlay this with the structure of the BP HKA, then you can see that we have a green side chain here, and that takes up the position where the phosphate sits in the docking experiment here, which would explain why the phosphate can't enter the active site in some of these pyruvate-dependent aldolases, while others that are more similar to the SW will be activated by phosphate. So it's the side chain that decides on whether you have phosphate activation or not. Good, so at that stage of our research, what we had reached was we knew the metal. The hydroxypyruvic acid coordinates like the pyruvate, and that was that phenylalanine that was important to get the allowance to get the hydroxy group in there. The arginine was essential in our case. The histidine was again the same as before. We now have an idea on why the phosphate acts as a cofactor, but what about other structural details? And by then, because we're trying to get that phosphate in so often, we had tons of X-ray structures. And if you look at the structures, then you've got a TIM barrel and you have a domain swap. And due to the domain swap, you've got this wonderful dimer. And the dimer then forms overall a hexamer. So 3 of the dimers form a hexamer. And here you can see one of these dimers, yellow and magenta. But the active site is actually, when you go back, is not sitting in the middle of the TIM barrel, what you would normally expect. But the active site sits between 2 domains, but not the domain swapped one. So the catalytic dimer is here, sitting here. So you see the hydroxypyruvate and the metal, and this is one monomer, and this is the second monomer. And we've got 6 active sites, of course, in this hexamer overall. And if you look at the metal, it's coordinated by this hydroxypyruvic acid. And when we examined all of the structures we had measured, we noticed something strange. And that was, if there was no hydroxypyruvic acid, the metal sat in a different position. And it was all of a sudden not only coordinated by those 2 acidic groups, as had also been described in the literature, but all of a sudden this serine from the other subunit also coordinated. So now we have a tri-coordinated metal here, 3 water molecules coordinated, also hexa-coordinated octahedron, just like here, but the shift here of the metal is significant. So the metal moves by 2.8 angstroms, and it can't be in both positions at the same time, so they exclude each other. So this came as a surprise, and then we looked at all the structures that we had with manganese and with magnesium, and what we found is that in the resting state, it's always coordinated to the serine. We found one intermediate state where you've got a pyramid. So this is the square of the pyramid, and this is the tip of the pyramid. And here it's again hexa-coordinated octahedral structure. We then did quantum mechanical calculations in order to check for the stability. So we take this as 0. For this here, it is indeed this structure that is more stable than the hexa-coordinated one. That would be a lot less stable. And if the pyruvate is coordinated, we go to a more stable structure. So it seems that this really is the favored structure, but you need the pyruvate to get it there. Well, this is kinetically favored, and we were first wondering whether it goes there in order to stabilize the structure, because it's at the interface of the 2 subunits. So we took the ARPO enzyme and looked at the melting temperatures. And we also had an alanine mutant for this one and a cysteine mutant. The cysteine mutant didn't express well and was less stable, but this is the same. You add the metal, these 2 are the same. So obviously this additional coordination does not stabilize anything. This one is slightly less stable, but it's also improved stability. And when you have the hydroxypyruvate, all of them show exactly the same stability. So we can conclude that this resting state is not there to stabilize the structure. And the dissociation between these catalytic sites is not the thing how it denatures. And most likely, we have a stabilization of the metal to prevent leaching between catalytic cycles. Okay, we then overlaid with the BP HKA to check, do we have the same thing happening there? And if you look at this, so that's the picture I showed to you earlier, the metal's essentially in the same positions, the hydroxypyruvates, the two acidic residues coordinating. If you then take the same enzymes, the X-ray structures, but without hydroxypyruvic acid, then for the SW, we see that we have a tri-coordinated metal. This is the serine from the other side chain. These are the two acidic residues. But for the BP-HK, we don't see the shift. We still have the two acidic residues coordinating, but the side chain here in green doesn't have a serine, and there's no movement whatsoever. If we look at an overlay of the pyruvate coordinated and the non-pyruvate coordinated BP-HKAs, we do see that the small shift of the metal does take place once the hydroxypyruvate coordinates, but the entire backbone doesn't move. So, to summarize all of this, in SWHKA, you have two distinct binding sites for the metal in this enzyme. You have a resting state and you've got the active state, and you've got a movement of 2.4 angstroms between them. Why we have this resting state so far is not known. For BP-HKA, we only have one binding site and a small movement during catalysis, which is something also known for other enzymes. Overall, for both enzymes, the entire protein does not move. It's only the metal that moves. The rest is completely static. But the most important conclusion of my talk is that if you work with metalloenzymes, don't assume the metal is completely stationary. It might very well move. And we did a thorough literature research, and we found that there are a few other examples where this has also been described, and we wrote a review about it. And I just checked, it's not yet online, so it should be online hopefully tomorrow or the day after, so then you can have a look there as well. But really the key message is metals in metalloenzymes are not static. And when you look at your mechanisms, look whether your metal actually moves and whether that influences the catalytic activity of your metal. Okay, I now come to the nicest part of my talk, which is the acknowledgement. Special thanks go to Stefan, who started up this entire area, and to Afina, who did the entire BP-HKA characterization. Sander had started it, but back then COVID hit and the project came to a standstill for a while, and Athena beautifully picked up on this. Laura produced tons of these enzymes for all of our different studies. Peter Leon did all the beautiful EPR studies. So that are all the people in Delft. Isabel Bento at the DAISY Embl lab did all the X-ray structures for us. And Hein Weimer from the Universiteit Groningen did the quantum mechanical calculations. And I'd like to thank every member of the biocatalysis group in Delft for the great working environment. And I'd like to thank you for your kind attention.
Thank you so much.